My name is Ross Jones. I work at the Australian Institute of Marine Science. I'm a coral biologist. I'm actually based in the Perth office. I happened to be in Darwin a couple of days ago, and I flew over for this just as a can you turn the volume up? Just dropping in. You can hear me back? So yeah, I'm a coral biologist. Because I've been based over in Perth, I've been a lot to do with something called the Western Australian Marine Science Institution Dredging Science Node. That's a huge $20 million research program, which is about exactly this issue. So over in West Australia, there's been some enormous dredging projects. It's all about predicting effects, characterizing effects, and the mantra of this $20 million program is to improve the ability to predict and manage dredging in sensitive marine environments. So it's all about characterizing pressure fields and relating that to the biology. This is an idea of some of the projects that have been going on there. That's the Pilbara region of northwest Australia. You can look at the size of these projects. These are massive projects occurring in very, very short periods of time, 1 to 2 years, $1 million a day dredging programs. And we have been actually involved in virtually all of these in terms of looking at sort of the effects that they can have on local communities. It's all reef communities. So I'm going to talk about the activities impacting on the sensitive receptors, and a lot of it's to do with dredging plumes. So this is from something called the Barrow Island project, and that is functionally equivalent to Heron Island or Lizard Island. It's a clear water environment. This is the worst case scenario. It was a 13 million cubic meter project that ran over 1.5 years right on the reef itself. So the plumes that you see, the dredging plumes, those are one of the concerns. With dredging, you have the direct effects, which is for capital projects is removing the seabed. You have the indirect effects associated with the migration of these plumes. You hear the word turbidity. It's effectively water cloudiness. And this is why there's a lot of problems with this and why it's controversial to do the dredging, is because the water quality, to be able to relate the water quality to what's going on on the seabed is difficult, because there are multiple different cause-effect pathways whereby sediments could affect corals, for this example. So you have turbidity, water cloudiness. Underneath that turbid cloud, it's affecting the light availability. Sediments are very good at decreasing the light, attenuating the light in the water column. If that plume descends back down on the seabed, you can then the suspended sediments in the water will then start interacting with the sponges and the corals and filter feeders. So you have effectively another mechanism that sediment can act on the corals. And a third one, if the sediment plume goes all the way back down, it can actually start smothering corals. So what you're seeing there is an extreme example. These images are from the Nelly Bay Safe Harbour. So that was Nelly Bay before. Afterwards, that was it before, and it's from actually dredging the entrance channel to Nelly Bay. So I was involved in that actually with Peter Ridd here for in about 10-15 years ago, or 2004. Those corals that I just showed were actually where the sediment spilled out the corner. So for context purposes, those are sediments, corals that got hit very, very hard by probably an accidental release of sediment. I want to try and work a little bit more on this concept of multiple cause-effect pathways. You've got suspended sediments, light availability, and sediment deposition on the apices of the triangle. If you're a sponge, you're probably most likely sensitive to suspended sediments. We know that they filter water all the time. If there's high suspended sediment concentrations, they won't like that. We know seagrasses are very sensitive to light. They need quite high light requirements. They live in the sand. Sediment deposition is probably not an issue for them. So their light availability is a key cause-effect pathway, the mechanism where sediment can have an effect. For corals, we're putting them right in the middle there. They're equally affected really by all of those different parameters as I showed previously. But it gets more complicated than that for the corals, because if we take an example like that, where the plume is going over the top, the coral is being affected effectively by light availability in that situation when the plume descends back down. And that's some of the images that you can actually see in these large-scale dredging projects. About 100 meters away from a massive dredge, it actually attenuates all light underwater. So you're seeing light attenuation. So the corals are being affected by light attenuation and suspended sediments. As it descends back down and you get sediment deposition going on, it's a bit of light attenuation, sediment deposition, and then again right up close to the dredge you get all of those parameters are all equally affecting it. And day by day, hour by hour, it could be different mechanisms which are affecting a coral or a sponge or a seagrass. They're continually changing. We call it the protein nature of suspended sediments. So you've got multiple cause-effect pathways. It's difficult to establish relationships to this. That can act alone or they can act in combination with multiple different types of organisms with different sensitivity. So it's a complex bag. We have, in Western Australia, managed to get all the water quality monitoring data from all of these projects. And none of this has really been published before a detailed analysis of water quality during dredging programs. So we managed to get information from 75 sites from different types of environments and look to see what is the sort of thing that goes on during a dredging program. You can see turbidity on the left-hand side, so the water cloudiness increasing when the dredging's starting, and you can see the light availability decreasing as the plumes move overhead. Again, you can see corals being sometimes put in darkness right up close to a dredge. I can't emphasize that enough. You're very, very close to the dredge. But you can also see at the bottom some unusual pigmentation, some green pigmentation, where the corals actually get put into a different type of light. Most of what I've said so far is written up in a publication, and especially all the changes in the water quality that can occur close to dredging projects. So this is just emphasizing this is quite well studied now in terms of the changes in intensity, duration, frequency of turbidity events, and what is a typical exposure regime. Prior to all this work being produced, we had no idea if you're 100 or 200 meters away from what type of dredge, what would be your likely exposure scenarios in terms of light reduction, sediment deposition, suspended sediments. We took all that information. This is the Barrow Island dredging project. This monitoring project cost $130 million. This is extreme, this one. It's because they actually decided to put an LNG processing plant on the reef itself. The EPA in West Australia said no. It got overturned at state and federal level, and they said no, it will go ahead. The monitoring that they had to do with it was absolutely excessive at 130 million. That's all the water quality monitoring sites, and you can see the plume effectively went south virtually for the whole project. And we've got this data, we've analysed this over about a 2-year period. It's an enormous data set, and we can actually start to define distances. So you've got distance along your x-axis, on your horizontal axis. You can start to see where it attenuates with time. So a lot of the way you handle these projects is that your dredging projects, you do have a zoning scheme, which I think Brad is going to talk about a little bit later. So you're interested in, right up close to dredging, what's your worst case scenarios? How does that attenuate with distance? And we're looking at the top there at turbidity, and at the bottom is light. So we actually quite rapidly have got a lot of information now about the likely exposure scenarios of these different pressure parameters during dredging projects. Just want to go quickly back to this one, which is basically a model of your light attenuation underwater. So on the left-hand side, it shows under different scenarios and under different suspended sediment concentrations, what your light levels will be. It's just a way of showing the attenuation that occurs. So under the orange one, the 10 milligram per litre concentration of light, at 5 meters, you're looking at 135 micromole quanta. It's a measure of light. If you're in crystal clear blue water, you'll be looking at 896. So you're looking at about a 5-fold decrease in light caused by quite a low concentration of sediments. The significance of this is also the changes in light quality. So this figure here shows the different spectral quality of the light. And the key thing is by the time you get down to a 10 milligram per litre solution, the suspension, all the light has actually changed into being an orangey green colour. So, sorry, a yellow-green colour. So what I'm telling you there is that the quality of light decreases as well as the quantity of light. And this is something that we're working on in Cleveland Bay, and I'll talk about that in a second. All of that's been written up in this paper. It's a public access, open access papers, and essentially I won't repeat it because I'll try and cut the time down a bit. One of the key pressure parameters associated with dredging is sediment deposition. And on the left-hand side, you can see some sediments that, again, from Nelly Bay that have been smothered in sediment. If you wash the sediments away, you can see it's killed the area. That, again, was about 100 meters away from dredging. What you see in the middle there is a new type of deposition sensor. It's actually been designed by Peter Ridd. And we've been using this in Western Australia, and a modified version of that we're going to get into Cleveland Bay very, very soon, or we've already had goes of it in Cleveland Bay. You see it on the top right-hand corner. It's in tanks. We're testing it. And what this effectively does is just measure sediment deposition. There's no technique for measuring sediment deposition at the time frames that you need in terms of milligrams per centimetre squared per hour or per day. So this is a new type of instrumentation which was built during the Wamsey dredging project, and we're about to apply that in Cleveland Bay, and that's the publication that's come from it. We actually deployed it in Middle Reef, which Peter just talked about a couple of minutes ago. These are probably some of the first measurements here of milligram per centimetre squared per day deposition rates on a coral reef. So it's one of the key parameters, and we think we've solved that to some degree. That's again going back to Barrow Island project. That's all the water quality that we've shown those slides before. Underneath each of those water quality monitoring sites, we have been given thousands and thousands, or hundreds of thousands of photographic images of corals as they go through all these different types of intensity duration of turbidity events. For each of those corals, they're taken at roughly at 2-week intervals, and we've been ploughing through this. You can see a coral that's been exposed, the sequences going from the top. This is over a 2-year period, all the way down. Corals, and there's a sponge behind it. That coral actually was probably 500 meters away from a dredge for a year and a half, and it was totally unaffected by the whole thing. It was the most perfect shape for doing it. It was a bell shape, so it could quite easily sweep the sediment off of itself. The sponge underneath actually fared quite well as well. It actually managed to be for about 6 months under sediment and wasn't affected by that. What we're doing at the moment is relating the water quality to what you can see of the corals and the mortalities of the corals, and we can derive water quality guidelines and increase confidence in the ability to predict and manage dredging from that. The problem with that is that we're actually doing it's a correlative thing. We don't know what's killing the corals or damaging the corals. We don't know if it's suspended sediments or light or sediment deposition. So, which is very dangerous if you don't know what the exact cause-effect pathway is. And that's what we're heading towards in the project I'm just going to talk about that we're doing with Port of Townsville. But how we overcome this problem of finding out what the mechanism is that we can do manipulative experiments. So this is the CSIM, which is the sea simulator. It's at Ames in Cleveland Bay, just over the other side at Cape Cleveland. This is top left-hand side. Well, it looks like a Bunnings warehouse, but it's a $30 million worth of research aquarium where we can really, really carefully manipulate all sorts of variables and recreate dredging events and dredging type water quality. So what you're seeing on the left-hand side is a lot of smaller containers, about 100 litres, which are highly replicated. On the right-hand side, much larger containers, about 1,000 litres, where you can put corals and sponges in there and start looking at the tolerance levels that they have to these different pressures from dredging. So we're looking at mechanisms and cause-effect pathways. That's 110 litres. So you can see some corals in there which are about to hit with elevated sediment. And these ones here are actually in the process of being exposed to suspended sediment concentrations. Those concentrations we're using, the 30, 110 milligrams per litre. We get that information from West Australia from all our analysis of the water quality. So we're doing environmentally irrelevant exposures. And then we're looking for various endpoints to see what the things are doing, what's happening to them. These ones are usually mostly associated with doing sediment deposition. So we have the deposition sensor in one of these tanks together with some corals. So we're looking at sediment deposition thresholds. Quickly go through these. It's just what we've been at for the last 2 or 3 years, and what we're carrying on doing is just lots of funny little manipulative experiments. So in the top right-hand corner, we increase the light levels to eliminate light as being a variable, and we can expose corals to elevated sediment concentrations. From that, we're actually figuring out that suspended sediments aren't really that damaging. It's actually the light attenuating properties of the sediments which is more damaging. We're doing experiments to see how long corals can stand extended periods in darkness, because that's a key variable for close up to a dredge, and then combined effects of elevated turbidity. Looking at sediment deposition, again, it's a coral from a picture from Magnetic Island. It's just off of Nelly Bay. The morphology of the coral really determines how well they can handle sedimentation. So we're doing experiments with different types of corals, looking at different deposition rates, different silicoclastic sediments, carbonate sediments, different sediment types, including some collected from Cleveland Bay. And we're looking to see the tolerance levels that they have to deposition that could occur during dredging. Coral bleaching on the left-hand side, you're seeing a coral bleaching event. It's elevated water temperatures. You've all heard of coral bleaching. We were interested to see whether the ability of the corals to deal with elevated turbidity is affected by bleaching. So we basically bleached some corals under temperature stress, put them through a whole dose-response series with elevated suspended sediments, and yes, it does affect their ability to process to clear sediment from their surfaces. Everything we've done, I've just talked about, has been about corals. We have replicated it entirely with sponges. So we're looking at phototrophic sponges which actually contain photosynthetic algae inside and bacteria inside it, and with heterotrophic sponges. And then we've done a whole complete sequence doing pretty much the same thing. And for the seagrasses, there's a lot of better information on seagrass in East Australia. This is some work that's done over in West Australia, which is again looking at the same sort of thing, minimum light requirements for seagrasses over during dredging programs. What light levels do they need so they won't fall over? All of this that I've shown you so far is on a WAMSI website. And it contains all sorts of news feeds and updates. You can get onto this via Twitter and Facebook. And all the publications that come out of WAMSI, there's 26 journal publications in the last 2 years. I can't emphasize just how huge that is. So 25 scientific publications in 2 years on exactly the issue that we're facing now. There's been a real step change in the amount of information that's available to be able to evaluate these projects and look at the impact prediction. There's a LinkedIn page. If you're on that, you'll get me, and I can sign you up, and I can give you all the different posts in the journal articles when they come in. So anything that I've talked about is available online. Now, I want to just quickly, how long have I got?
4 minutes.
4 minutes. So, whilst we were doing this stuff in Western Australia, I actually happened to meet Melinda in a conference at a workshop, and I said, look, one of the biggest problems we have in Western Australia is we can't get up close to the dredges, and we want to look at the effect of sediments in turbid water environments such as Cleveland Bay. We got some money from Commonwealth Government in terms of something, the National Environment Science Programme, to look at, well, you've seen that picture before, so we'll cut away, but to look at turbidity in turbid environments and to use everything that we'd learned from Western Australia and apply it into this situation in Cleveland Bay. What Port of Townsville gave us was access to their boat and allowed us to actually get right up close to the dredge, so we won't have a 500-metre exclusion zone, which we previously had. And we've been doing the similar sort of things that we did in Western Australia. We're getting profilers, looking at light quality and quantity profiles in Cleveland Bay. There's some stuff there just way north of here, the 18-metre contour. You have looking at the light spectrum through clear water, very, very high levels in blue. It attenuates with depth. If you go back into the turbid water in Cleveland Bay now, you can see the similar sort of effect where the green, that you lose your light, but the spectrum changes. What we're doing is in the CSIM at Ames, we're trying to replicate what we've got so that we can recreate the conditions. That's the one in red is Cleveland Bay, the one in blue, yellow is from the CSIM. So these are different LED lights. So we can pretty much replicate the light conditions. We're missing a bit here, and that's because we need to get an orange LED light in there from China, which is under order to try and... So what we're going to do on the right-hand side is to replicate the conditions in the tanks, and then go through the whole sequence of looking at filter feeder communities, corals, and also coral larvae. We've got a big program starting looking at juvenile corals, effect of sediments on juvenile corals. We'll be deploying the deposition sensors of PETAs. We've already got that going in there, so we can start characterizing the natural background sedimentation rates or sediment deposition rates in turbid environment. And we've also been, Port of Townsville gave us all of their water quality monitoring data, so they've got 4 different sites. There's also an existing monitoring program with Ames and Gabrumpa, and we're going to have the data for that, and we can actually start characterizing what is the normal turbidity regimes in Cleveland Bay, and then use that information to derive thresholds. So been asked to cut it down a bit, but in the last 30 seconds, just reflecting on this, there has been an enormous amount of work in exactly the issue that you're facing here through this work in West Australia. The funding for that came from an environmental offset, so it was the industry that had to pay for that. for that program, and it was co-investment from people like Ames and CSIRO and JCU and all the universities over in the West. The amount of information coming up is just extraordinary, and one thing I heard this morning is that the framework that they are using for the EIS for Cleveland Bay is based on a zoning scheme, which is the West Australia scheme. So they've adopted the West Australia scheme. So all the research that we have been doing can feed directly into that. Where we're heading with this, as I said, is looking at different species, different sensitive, different life stages and so on, and trying to actually develop threshold water quality guidelines which are local to Cleveland Bay, with the intention that somehow these get incorporated into monitoring programs. I'm node leader for science of that program in Western Australia. There's a co-node leader who's the node leader for policy, and that is the guy who devised these EAG7 guidelines, which is how you do these projects. So I'll wrap it up there just to say that the difficulty is that there are multiple cause-effect pathways. A lot of them haven't been characterized, they have been now. Some of them are difficult to measure, like deposition, but we can effectively measure deposition. And the difficulty is why this hasn't been solved before about what are the impacts that this will have in being able to predict and manage is, say, there are multiple ways that the sediments can affect organisms, and it's a complicated process. Thank you.